I'm Yogesh Pai, Assistant Professor of Law at National Law University, Delhi. Today we are going to discuss a module on biotechnology patents, subject matter exclusions. So let me briefly explain the learning objectives. So we are here to understand how modern biotechnology has helped markets in bringing out innovative products that are very technology intensive, to understand the role of patent in incentivizing biotech inventions and the challenges that biotech technology poses to the patent system, to examine the early origins of patenting biotechnology subject matter in different jurisdictions and in India, to also understand how patent eligibility of biotech subject matter has been discussed and debated during the TRIPS context, and also what was the outcome in the TRIPS negotiations in the form of Article 27, and finally to examine the provisions in the Indian Patents Act 1970 on the question of patentability and patent eligibility of biotech subject matter, specifically in the light of the biotech guidelines issued by the Indian Patent Office in 2013. So first question that we need to ask in the context of modern biotechnology is, what is biotechnology? Any technological application that uses biological systems, living organisms or derivatives thereof to make or modify products or processes for specific use is what has been largely defined as biotechnology, and this is a standard definition that flows from Article 2 of the UN Convention on Biological Diversity. The second important point to note in the context of biotechnology is all DNA is made up of a base consisting of sugar, phosphate and one nitrogen base. There are 4 nitrogen bases: adenine, thymine, guanine and cytosine, and this is popularly called as ATGC. These nitrogen bases are found in pairs with A and T and G and C paired together. The sequence of nitrogen bases can be arranged in many infinite ways, and their structure is known as popularly called as the double helix. It may also be noted that the 4 nitrogen bases are the same for all organisms. Hence, discovery of recombinant DNA technology has changed everything. Recombinant DNA, or what is called as rDNA, involves taking one strand of the DNA and combining it with different strands of DNA to effectuate a desired particular result or a trait. The particular figure explains the difference between biotechnology as a whole and modern biotechnology as a subset. So in biotechnology as a whole, you have natural regeneration, cultivation methods, tissue culture and natural breeding. And in modern biotechnology, you have cell fusion, transfection, transformation, conjugation, agroinfection, and in case of high life forms, you have the GNOs, in vitro manipulated through DNA and RNA. So what are these biotech products ultimately? So modern biotechnology has applications in several areas, including but not limited to medicines, agriculture and industrial purposes, largely to include industrial fermentation, etc. So in case of GMOs, the classic examples of GMOs or genetically modified organisms are Harvard Onco mouse, Oxitec mosquitoes. So these are high life forms that have been genetically modified. In case of medicines, you have classic examples like Rituxan, which is produced by Genentech, Humira, which is produced by Abbott, Avastin, which is produced by Genentech again, Herceptin or trastuzumab as it is popularly called, which is a breast cancer drug produced by Genentech, and Epogen produced by Amgen. And there are a host of other products that have been invented in the last few years which are based on biotechnology. So what are the type of claims that are usually made to the Patent Office? The Indian Patent Office has defined or provided a brief description of these guidelines of these such claims in their guidelines in 2013. So they say that the biotechnology applications comprise the claims relating to certain subject matters like polynucleotides or gene sequences, which may either be product or process based claims, polypeptides or protein sequences, again which are product and process based claims, vectors like plasmids, which may be in the form of product or a combination of product and process claims, gene constructs or cassettes in gene libraries, host cells, microorganisms and stem cells, which may again be in the form of product and process claims, transgenic cells, plants and animal tissue culture, pharmaceutical or vaccine composition comprising microorganisms, protein, polynucleotides, which are again in the form of both product and process claims, antibodies or antigen binding fragments thereof, which can be monoclonal antibodies or polyclonal, diagnostic kits and tests, diagnostics tests such as the tests for detection of a mutation in a gene, protein which might be associated with a particular condition such as protein expression or a disease. Now, these diagnostic kits or diagnostic tests can also be in the form of process claims, and as we shall see from cases, these claims have been heavily contested in different jurisdictions, and the Patent Office has made an humble attempt to come out with certain guidelines which provides a framework for biotech applicants in India. So what are the challenges that biotechnology poses to the modern patent system, or how modern biotechnology challenges the traditional patent law? Advance in sciences has now made it possible to isolate and replicate a host of a naturally occurring substance, and thus it raises questions for patent law, and those questions can be like, can it be shown that when viewed as a whole, an application incorporates a discovery that brings about a technical change, and should it be patentable? Second, finding of a new property of a known material or article to be treated as, should it be treated as an unpatentable discovery or should it be patentable? Whether use of such properties to a practical use, may that be patentable or should you need something more? The difficulty of adjudging what forms invention vis-a-vis discovery in biotechnology, specifically in relation to patents on gene sequences or express sequence tags, primarily the arguments are that there is a human intervention criterion. Both discoveries and invention involve human intervention, involve time, labor, skill and investments. How do you distinguish? We thus need to ask the question, what type of human intervention is demanded for making unpatentable discovery and invention? Guidance can be taken from patents that have been granted. It is otherwise too difficult to draw a plain line of difference and arrive at a straitjacket formula. The Indian Patent Office has issued a manual published, and it has also issued the biotech guidelines, so that may be one of the important ways to find out what is or what is not patentable, especially its distinction between invention and discovery. The Indian Patent Office has said that there is a difference between discovery and invention. A discovery adds to the amount of human knowledge by discussing something already existent which has not been seen before, whereas an invention adds to the human knowledge by creating a new product or a process involving a technical advance as compared to existing knowledge. The emphasis seems to be that there needs to be a new product or a process which represents sufficient technical advance when compared to existing knowledge, and something that is already known before or seen before cannot be patented.